We'd introduced the anatomical position, and this is consistent so that naming makes sense, describing the structure and function, orientation of organs within the body makes sense. So you can convey that to other anatomists or physiologists. We identified that while this is described standing up, if you lie down, the position holds up, the features, the directional terms are still intact. It's all in relation to the the organs within the body as a whole. Now the body itself is large, and so we break the body down into individual regions, which help to describe features of those regions or the location of interest. Common ones that you'll know are the head and neck, the trunk, upper limbs, lower limbs, but even those are a fairly large scale. There are a lot of individual structures within those areas, and they're subdivided further as outlined here. And now these will become more clear as we go through the material. We often use names of the regions or portions of the names of the regions to describe nerves or muscles or bones. So it will become obvious what regions we're referring to as we learn those names as well. But this is a very good starting point. You can start by memorizing the slide if you want to, and that will help you understand the names of nerves and muscles, etc., when we get to them. But we will explore these in depth as we move through the course. If you don't know all of the individual regions, you might be able to describe the feature you're looking for using directional terms. If you know one main region, you can describe the relationship to that main region for a certain characteristic of interest. These allow us to precisely locate features in the body, and they're all relative to something. So you do have to have a starting point, and then the directional terms will tell you where to go from that starting point. They usually occur in pairs, and they're usually opposed. So proximal and distal is a pair, referred to towards or away from the core or the trunk or the center of the body or system. Proximal or distal. Medial and lateral are ones you've probably heard before. Towards the middle, medial, lateral, away from the middle. We often use superior and inferior to describe near the head or towards the feet. This will still hold up if the person, the body, the subject is lying down. So lying down horizontally and saying something's inferior or you're looking inferior to a certain process means towards the feet. So in this example, the radius, for instance, is distal to the humerus. That makes sense. That's how you would describe it. The ulna is medial to the radius. It's towards the middle of the body, even though it's in the upper limb on one side. The ribs are superficial to the lungs. They encompass the lungs. They're towards the surface of the body, not deep to the body. Superficial versus deep is another pair. You could say ribs are anterior and lateral and posterior because they encompass the lungs. But really superficial is a clean, easy way to get there. All of the pairs are shown in your book, and the ones that we've highlighted or common ones that we've looked at are shown here, but they all will be used to describe the position of features or structures within the body. Some are more complicated than others, and we'll get to those in turn. So you should know these. Take some time to review the table. when we describe things in lab, we might just use these terms off the cuff and expect that you know what they are. I do a lot of work with dehydration in my research, and we collect samples of sweat, and I'll often use a little bandage or what I call a sweat patch. And we affix it to the body to collect a sample of sweat while a person's exercising, and we do other stuff with it later. But the positioning of those patches uses or relies heavily on these directional terms. So you can say put a patch on the chest or a patch on the forearm, but that doesn't tell you very much. If I say at the midpoint of the clavicle along the inferior border in the right pectoral region, you know what I'm looking for. You know that general area that I want to apply a patch to. Or the midpoint of the forearm on the dorsal aspect of the forearm along the midline of the right forearm. It could be either. You know what I'm looking for. So the directional terms help us to map and landmark the body. We'll often also not look at the body as a whole, but we'll break it into sections. And if we think about one upper limb or one lower limb, you know what to think, you know what to picture. But planes will allow us to see through parts of the body to internal structures that might not be obvious at the very superficial levels. So planes and sections are windows to those internal parts of the body. They're literally like panes of glass that cut the body or a part of the body into sections, and then we view that facing surface and all the internal aspects of that organ accordingly. So common planes are shown here. A sagittal section cuts the body into left and right halves. If the left and right halves are exactly equal, it's a special kind of sagittal section called the mid-sagittal section, right down the middle of the body in this case, or it could be an organ. A frontal plane or a frontal section breaks the body into anterior and posterior sections. The frontal plane is shown here. Transverse plane breaks them into upper and lower, also referred to as a cross-sectional or horizontal plane. And then anything else we call an oblique plane. Anything else at a weird angle, not along one of those axes, is an oblique plane. I think we like sticking to the main sections because we know what to expect along those main axes. Oblique planes give us some good information, but they're sometimes hard to orient yourself to what structure or where in the body you're looking at. Anything else? So maybe this is review, but let's practice briefly. We've done it at the body level, the whole body level. We can use an organ such as the brain to do the same thing, to section and then view the internal structures of the brain. So we have a section at the top going through the exact middle of the brain, splitting it into left and right halves. The mid-sagittal plane allows us to look at the midline outwards. And we see that here on the right-hand side. Mid-sagittal section created by the mid-sagittal plane. In the same way, the frontal plane will create a frontal section. And notice the view arrow in each of these examples shows us which way we're looking. Because in 2 dimensions, if you don't have that orientation, it's impossible to know if you're looking forward or backwards. And that doesn't often cause problems when the organ is mirrored. It might cause problems if you don't know which way is left or which way is right. We'll see examples of that later. And then the cross-sectional or transverse plane at the bottom, we are looking down from the top of the skull, or the top of the brain. So this is looking down on the lower inferior portion of the brain. Transverse section shown at the bottom. It's easy with all these examples on the screen. I happen to have sections of my own brain. In 2015, I was involved in a pretty bad bike accident, major concussion. I don't remember what happened for half an hour after it. I got to an ambulance, someone called an ambulance, didn't make sense, and I woke up as they were asking my phone number and address and name on the way to the hospital. So I was recruited after that event to be part of a concussion study at Sunnybrook. Had definitely a major concussion. And did a few MRI scans. The details of the study aren't necessarily important. MRIs, though, are very confining. I realized I didn't like that when I was in the middle of one. But these are sections of my brain. So we have one slice here. You can see some features of the brain that we'll get into in more detail as we get into that section in 1 or 2 weeks. It's coming up quick. You can see the eyes, the brain stem, something called the cerebellum. What section is this? Transverse. Okay, which way am I looking? Up or down? Are you sure? I'm not sure. You could be looking up. It's impossible to tell in 2 dimensions. Definitely, transverse plane. Fantastic. Probably looking down, if we think about how an MRI might work. Superior view. This one? What section is that? Did I hear sagittal? I didn't hear sagittal. Good, because that's wrong. Yeah? Medial. Medial. Well, of the ones that we've talked about, medial would be down the midline, and the orientation we haven't defined in that context, but we didn't mention medial necessarily right now. Maybe it's down the middle, splitting the brain into 2 halves. But what plane that we discussed would you suggest this one is? Not medial. Frontal. Splitting the organ or the body into anterior and posterior halves, or sections, front and back halves. Frontal plane. You can see something called the ventricles there. That might give us a hint as to whether we're looking forward or backwards, whether the ventricles are larger on one side than the other. That's one defining feature that might help us orient ourselves. We'll see that coming up. And then, of course, the last plane, sagittal. This might be a mid-sagittal plane, a medial plane. If it's directly through the 2 hemispheres of the brain, splitting them exactly in half. Sagittal plane, it's probably close to the midline. There should be a little membrane here that separates the 2 ventricles. That usually is in the midline, so I'm not going to say it's exactly in the middle. Or maybe that membrane for me is a little bit off to the side, I'm not sure. I'm also concerned about like some of the spacing. I didn't realize I had a pointy posterior part of my skull until I saw this. And there's some space, and I hope that's not atrophy, degradation of the brain tissue. But easy way to practice planes, which is good. Probably mid-sagittal, very close, if not exactly mid-sagittal. So we have ways to describe how to get to features within the body. We have regions to describe where we're looking within the body or on the body. Body cavities are another way that we can describe our focus. They're like regions, but they describe the container or parts of a container. Usually these cavities will contain major organs that often work towards a common goal. We use these to describe sections. Here we have the torso, broken into many different chunks or cavities, that each contain, let me not continue with that. I'm going to confuse you. For instance, if we look at the cranial cavity and the spinal cord, the yellow highlighted sections in the middle, they are connected and they contain the central nervous system, the brain, and the spinal cord. They're covered with layers of protected wrapping on the insides to protect the contents. We call them meninges, and you'll see all about those coming up. The thoracic cavity is the area bordered by the thorax, the sternum, the ribs, the spine, and at the bottom by the diaphragm. And that cavity is large. It contains many important features. And even that is too gross a level of overview. We even break down the cavities into subsections or subcavities. Notice here we mention pleural, pericardial, and the mediastinum, which we can see on this next slide. So the thoracic cavity we can see is broken into multiple different cavities. The pleural cavities are right and left. They each contain one lung. The mediastinum describes the middle section between the lungs. And the pericardium, the pericardial cavity, contains the heart specifically. This is another way that we can break down regions within the body and then identify where we are looking to describe certain features. These subcavities are almost just thin layers or thin cushions. They are the red and the pink sections shown here around the heart and the lungs. And then the dashed bordered outline indicates the mediastinum. The diaphragm at the bottom is the lower support or the lower border of the thoracic cavity, but the superior border of the abdominal cavity, or the abdominopelvic cavity. And there is much more detail about these sections in the book. I don't think it's important to necessarily divide into quadrants the abdominal cavity and look at how we name all of these features. That is available for you to look over, but all I want to identify here is the type of structure and the fact that we can subdivide the cavities. Well, what subdivides these cavities? What are the borders of these cavities? A special type of what we'll call epithelium that we'll get into a bit later in the class divides these cavities, or sequesters, breaks the cavity up into multiple sections. We call these serous membranes. They are generally thin, double-layered membranes or double-layered cushions that separate the cavity into smaller subsections. They line the walls of the cavity and they line the organs themselves, and depending on which surface you're referring to, it has a different name. The visceral layer is what borders the internal organs, or the viscera. The parietal layer borders the inside of the cavity itself, or the surface, the walls of the cavity. And the visceral and parietal layers are outlined for the pleural cavity on the left, the pericardium you see extending over to the right. And you can see it points to the border immediately adjacent to the lungs in the case of the visceral pleura. And the border immediately or intimately related to the surface, or the internal surface of the cavity, the parietal pleura on the left-hand side. Now, the purpose of these membranes is mostly to allow movement, reduce friction, and prevent damage to the internal organs. They're generally closed. They contain some lubricating fluid that allow sliding of the two surfaces against one another. And imagine for the lungs inflating and deflating, the degree of stretch and friction that would be applied from the visceral to the parietal layer if there weren't some lubricating fluid between the two. We won't go into too much depth. That fluid we typically call serous fluid just meant to lubricate. Everything is closed as a cushion, allows beating of the heart, movement of the heart, inflation of the lungs, prevents friction. So we've oriented ourselves. Now, I want to talk briefly about characteristics that make a living organism. And this concept is one we return back to fairly often. The idea of balance, the idea of equilibrium, and the processes that allow the equilibrium to exist, for the body to function, for us to sit here, take notes, for me to lecture, for us to play sports. Characteristics shared by living organisms. Living organisms are differentiated from non-living by a number of different processes, and this garish background is simply to highlight the fact that there's usually a range in which we want these processes to occur. There's usually an ideal. And it's not so well defined what the limits of that ideal are. We know that as we venture away from whatever the ideal happens to be for one of these processes, we often don't want that adventure to be too long. We want it to be short-lived. We want to return to the normal situation. We'd like to stay in the middle of this green band if possible. So these life processes are metabolism, which we think of as the sum of all chemical processes in the body. It has two typical branches, anabolism and catabolism. Anabolism being the processes that create or build. Catabolism being those processes that break down or degrade. And we'd like metabolism to be in balance. If we think about tissues, building up and breaking down tissues, if those weren't in balance, we would end up with tissues that are much too large or no tissue. We want there to be balance. Responsiveness is a characteristic that allows us to detect changes in that balance. It could be in temperature. It could be in sounds that are nearby. It allows us to compare the current status with some set point or ideal, what we think should happen. Movement is one of the ways that we can react directly to that responsiveness. So at the largest scale, if we hear a predator, we can move to remove ourselves from harm's way. Not a problem anymore. We don't hunt saber-toothed tigers or woolly mammoths. We're not really in danger for a large portion of each day. Movement can also refer to molecules within the cell, or cells along a membrane, or the process by which we're able to grow, which is the next point. Cells differentiate and move and proliferate, allowing growth, which is another characteristic. The increase in size and form and function of body tissues, or the body as a whole. And really, growth is harnessing your environment to achieve that end. You don't just grow automatically. You need energy from somewhere, which happens to be in your immediate environment. You need to sense that energy and act on that energy to be able to take it up and support those growth processes. Differentiation describes that there are different cells to perform different tasks, or different organs to perform different tasks, like we have police officers, firefighters, professors, bakers, etc., different tasks to support society at large. Cells within the body will support the operating of the organism at large, and then reproduction we often think of as creating a new human, but this could also be repair or replacement of damaged cells, or DNA, which is an ongoing constitutive process in the body. without which we wouldn't make it much past a few years old. So all of these need to be intact and working properly for life to continue, quote unquote, normally. So that normal situation, that balance that I'm referring to, is what we call homeostasis. You've probably heard of homeostasis before. That equilibrium point where everything is in balance is what we are describing when we refer to homeostasis. It can be equilibrium in any number of aspects within the body. It could be the anabolism-catabolism example that we just talked about. It could be equilibrium in changes in pH. The body can't be too acidic or too basic. It could be changes in temperature. It could be changes in substrate availability, so carbohydrates, fat, protein. Hormones within the body, blood pressure. Blood pressure is a fantastic example. We often use that to describe one of the feedback loops. I think we do use it to describe the feedback loop coming up. Any number of processes are subject to this idea of equilibrium. And we see that illustrated here. So this is a trace of temperature over the course of a day. It's albeit perhaps a bit exaggerated and a little uniform. Here, over the course of a day, this person saw regular increases and decreases, or an undulating temperature response. And there's some ideal temperature in the archaic imperial units here, 98.6 degrees Fahrenheit. We like to think of, in terms of Celsius, 36, 37 Celsius is what we in the enlightened world like to use. There's some set point around which we would like to operate. Temperature is really important to maintain normal functioning of the enzymatic processes in the body. Higher temperature, they work too fast. Colder temperature, they work too slow, which is good for fridges, but not for us. And so there is some mechanism or response, or perhaps it's accidental, that every time temperature strays away from this equilibrium point in the middle, it ends up returning towards normal. And it overshoots, but then it's corrected. And the net average is that we live around what we call a set point. That is equilibrium. That's the ideal point. Here the set point is 98.6 degrees Fahrenheit. The set point for blood pressure would be something different. So this idea of homeostasis can be thought of on many different levels, on the systemic level, the organism level, or the cellular level. And on the cellular level, that really underpins homeostasis in an organ or a tissue. It's really dependent on the immediate environment, which in this case is described by the fluid or the connections to other cells or surrounding that cell. If cells generate waste products, they need to get rid of it. If they need fuel, they need access to it. And generally, the extracellular fluid is what will allow that exchange to occur. Extracellular fluid is simply a fancy name for fluid that is outside of the cell. Intracellular fluid, then, is fluid within the cell. All of the fluid in the body is either intracellular or extracellular. And so we can imagine, a good example here is the exchange of gases within the body. We know that we need to take up and consume oxygen to live. We end up producing and need to expel CO2 so that it doesn't build up in the body. And in large part, diffusion through the extracellular fluid is how that is accomplished at the cellular level. There are other complex factors involved. We'll talk about them next semester. There's a whole conduit system involved in that process. But from a cell's point of view, the fluid is where those gases are dissolved into and out of, where that exchange occurs. I also like this figure because it does a good job of showing the body in its true form as a donut. We're not bags of cells with fluid encased in a membrane. We're really donuts. And that's highlighted nicely by the digestive system over on the right. There's a clear opening and a portal all the way through, a clear opening on the other end that excretes solid waste. Anything in your digestive tract is not really in your body. Your body surrounds it. It's intimately related to and in contact with, but we're really a donut. We're not a cylinder. So this shows that nicely. And you can see the diffusion of nutrients or substrates, fuel, what have you, from that digestive system through the extracellular fluid by way of some roadmap towards the cells in the body, and the exchange reverses to get rid of any waste products. Also notice the subject of our next discussion, the integumentary system, contains, encases, and protects that internal system. I think it's important at this point, since we've introduced the idea of body fluids, to just briefly expand on that. This slide is not in the slides that were posted on Moodle. I just want to put into context the amounts and relative distribution of fluid in the body. All fluid within the body is either intracellular or extracellular. It's either in a cell or not. That makes sense. If it's neither of those, it's not in the body. We can see a small difference in the proportion of fluid between men and women, simply due to differences in tissue size. Women tend to have a higher percent body fat, and that simply displaces some of the fluid or decreases that percentage. But of the fluids, roughly 2/3 is inside a cell, 1/3 is outside a cell within the body. And so what we were talking about in the last slide is that 1/3 at the bottom, the extracellular fluid. The large majority of extracellular fluid is just between cells. It's in a tissue, it squishes between cells. And the technical name for that is interstitial fluid, which really just means between cells, fluid between cells. It is worth mentioning, and this will become clear later, that there is a small percentage that's not between cells, and that makes up the blood. Blood is in large part fluid. The majority of blood is fluid. It's not contained within a cell. It's not between cells, so it's not interstitial, but it is extracellular. That is the plasma. Interestingly, red blood cells that are transmitted by the plasma, the fluid within those cells is intracellular, just to confuse things even more. We'll come back to that in more detail when we get to blood as a tissue and the cardiovascular system. I thought it was worth mentioning now. So this framework supports the free exchange of oxygen, waste products, nutrients that allow us to control homeostasis. Homeostasis is that equilibrium, that ideal set point. And as much as we think we're always at that normal level, we're not. Homeostasis is always being disturbed. Temperature is not always ideal. pH balance is not always ideal. Moods aren't always ideal. Sleeping patterns are different. Food, eating patterns are different. I will, in large part, this is due to various stresses on the body. So the sun coming out today, which is fantastic for Nova Scotia. applies a heat stress to the body, and we have to regulate within that stress. The book starts to talk about, or mentions briefly, the idea that you don't have enough oxygen for a 2-mile run, and that's unequivocally wrong. There's never a lack of oxygen. You'll learn more about that in XPhys, but that's not a problem. Whether we can use that oxygen might be a problem, but there's always plenty of oxygen available in normal conditions at sea level like we are now. Even at altitude, there's enough oxygen. And we can talk about that later, but that's a more advanced topic. So these imbalances occur from disruptions like the sun coming out, like extreme temperature, and we need to respond in some way, shape, or form. This slide is included in the slide set that's on Moodle, and each of these is a link to some interesting animations. I'm not going to go through them in depth now. We do cover the same information that they do, but it's a nice refresher if you want to look at them on your own time. It's delivered in a slightly different way than I would deliver it. It's the same information repackaged. It might help you come to grips with the concept. The way that we respond to these stresses is a two-pronged approach. We have immediate response, quick or acute response, which is typically carried out by the nervous system, and then a more prolonged response through the endocrine system or through the release of hormones. And this is often a strategy that the body uses to have immediate feedback and then a more prolonged, sustained feedback or response. And we see it first here. So the nervous system has the beautiful capability of being able to react quickly, turn things on right away, while the endocrine system relies on the distribution of hormones through the blood, which takes a bit longer. So the control of homeostasis is outlined on this flow chart. We have some external stimulus at the top that affects whatever our controlled condition is, or controlled variable is another way to think about it. And every response needs to have three elements: a receptor, an effector, and between those two, a processor. So the variable in question needs to be sensed. We have to be able to monitor it to know whether or not it is at that set point or in balance. It's the job of receptors to do that. The controlled variable must be sensed. If we have that sensory information, receptors end with yes or no, this is where it is, that's it. That sensory information needs to somehow be interpreted, be compared to normal. The processor is what handles that comparison. The status right now is compared to some normal table or a set point that we have stored away. How we store that away is another question. But the condition of the immediate environment is compared to what it should be. If I'm outside in the sun and it makes my body temperature go up, I need to compare that to what it should be. When that comparison is made, if there's a deviation, we need to change it. The effector is what changes it. Action needs to be taken to correct any deviation from that set point, up or down. Usually the action is proportional to the deviation. If it's a small deviation, the response is small. If it's a big deviation, the response is large. This creates a new state. After the response has happened, our controlled variable is not in the same position or orientation or under the same conditions that it was sensed. It's now in a new state, and the process starts over again. So the signals that return to the processor that the receptors sense are called afferent or afferent. I like to emphasize the afferent, because the comparable signal on the output side is efferent or efferent. Afferent, easy to confuse those two. Afferent, efferent, a little bit easier to distinguish. Afferent signals go towards the processor. Efferent signals execute a task, move away from the processor. So in practice, all of these things act to correct a disturbance. And the majority of feedback loops, like we just described, are negative feedback loops. These are by far the most common within the body, where some feedback is sensed, an action is taken to correct and restore a disturbance. Now I'll contrast that with positive feedback, where an action is taken to strengthen a disturbance. A runaway freight train. That's what a positive feedback loop is. So negative feedback loop, by far the most common. Blood pressure is one of the go-to examples to illustrate how that works. So again, we have the same layout. There's some stimulus. You find out that there's going to be a pop quiz at the end of class. Stressful stimulus. All the heads looked up. That was great. There's not. But your blood pressure rise for a second. Right? When that happened, there are receptors in your body that sense that. We call these baroreceptors. They're specific to the stretch of the arteries. They're located in some very specific locations in very important arteries. And they always fire. They're always sending signals. And they send signals at a rate that is proportional to the stress. So when you thought we had a quiz, the stress increased, the stretch increased, blood pressure went up, and the firing of the baroreceptor increased. That signal, there's a deviation, pressure's higher than it should be, was sent to the processor. The processor here, for our purposes, is the brain. We'll get into more detail later. That's clearly a gross overgeneralization. But the processor in the brain interpreted the firing rate, says, hey, this is too fast. I need to consult my response table and figure out what do I do to return that firing rate to normal. It created a plan to return that firing rate to normal. That plan involves not pushing as much blood into the vasculature. And what I mean by that is it tried to slow the heart rate and expand the vasculature to decrease blood pressure. If you think about it, blood pressure is just, I'm a section of blood moving through the artery, and I'm pushing on the walls of the vasculature at all times. Walls of the arteries are containing the blood as it moves through the body. If I push less blood into that same small space, the outward pressure is lessened. If I do that less frequently, the pressure is lessened. So that plan created a new response, a new situation, where after blood pressure spiked from that stress, it has been reduced. Now you're back, hopefully, to a normal, bored, sleepy blood pressure that's much lower than it was before. All told, relaxation of the system reduces blood pressure back towards your set point. The text on this slide is new and not on Moodle, but you will see the animation. Oh, It's on the next slide. The animation that I have here highlighting a positive feedback loop is there for you to look through as well. Same example that we'll discuss briefly here is highlighted in those videos online. And we have 5 minutes, so I just want to briefly illustrate this. While the idea of a feedback loop is fresh in your mind, so a negative feedback loop corrects a deviation, counteracts a deviation. A positive feedback loop reinforces or magnifies some deviation. It doesn't return to a set point. It worsens or it magnifies the distance or the deviation from a set point. It continues until it's disrupted by some external factors. A common example here is childbirth. Classic example of a positive feedback loop. So when a baby is about to be born, uterine contractions seat the baby at the cervix. Press the head of the baby into the cervix. Now, the sensation, the receptors are in the cervix monitoring the stretch, a lot like the baroreceptors are. They relay that stretch information to, again, the brain, to some processor, saying there is stretch here, but this specific loop doesn't create an opposite response, it magnifies the response. The response here is to release hormones, activate the endocrine system, to release a very specific hormone called oxytocin, which travels through the bloodstream to the uterus to engage the muscles of the uterus, to strengthen the uterine contractions, to reinforce that feedback, to create more pressure, to create more stretch, to help give birth to the baby. That process feeds back on itself, magnifying every time the cycle completes until the stretch isn't there. Until the stretch is released, removed somehow. Usually, that is birth of the baby. Baby is born, exits the birth canal, stretch no longer registers. The positive feedback loop disengages. Much less common in the body. Positive feedback loops are much less common. Let's leave it there for today. We'll come back tomorrow at 1 o'clock. We'll continue and finish this section. Probably get into a little bit of integumentary stuff. Have a wonderful day.